So I'd like to introduce our next speaker, Dr. Giannoula Klement, who is a physician scientist. She's co-director of the Precision Medicine Program, as I had alluded to, and also chief of academic and research affairs in the division of pediatric hematology oncology. So she is a pediatric oncologist, but I think fair to say precision medicine is agnostic of age. In fact, she will be starting a precision medicine clinic in our adult cancer center in a few months. So we've asked her to speak about future paradigms for precision oncology.
A lot of what I'm going to present is from a publication of the same name that came out in May. But what I would like to do is give you a clinician's view. Much of what was discussed during the lunch discussion was you present clinicians with all this genomic and proteomic information, and most clinicians, unlike the scientific audience right now here, haven't been trained. Disclosure, much of the research for this presentation has been done with CSTS Healthcare, which has funded the scientists that have done the mathematical analysis. I have to say that in clinics, and that was acknowledged during that lunch panel discussion, we have had great successes with traditional chemotherapy. And I hope you notice that, I don't know if this points, the survival rates are in the 90s for acute lymphoblastic leukemia in children, and Wilms tumors in the 80s, rhabdo, not so great, but still better than it was in the past. Now, I put 2003 as one of the turnover events, which is when the 40 years of molecular research has actually trickled into the clinic with the approval of the first biologic agent, which was Avastin. The survival until then was actually quite low with most agents, and most of the improvements were truly supportive care. We still aren't doing great in some of the solid tumors. So while one can assume a linear relationship between dose of chemotherapy and cancer kill in leukemias, because of the stromal elements that help cancer cells survive in solid tumors, we have not had as much success with chemotherapy at maximum tolerated doses in tumors that are solid. And I have to say, and this is purposeful, 80% of our patients with PNET or Ewing sarcoma die on standard therapies. And we have to begin thinking that some of those 20% survival rates may not be due to our chemotherapeutic intervention. And those may be the the people or the patients that may benefit from targeted therapies. So I have to say that in 2003, with the introduction of first biological agents, have changed, and that more and more therapeutic agents are now entering clinical practice, and we do have to get used to how to use them. It is very difficult to keep up, though, with all the emerging information. I think it's much easier as a scientist. For many years, I have run a lab, and I realized that there's way more reading happening in a laboratory and in journal clubs than there is in clinics. The mechanisms of these agents are entirely new, and the science that has created those molecular agents, it's entirely unfamiliar to oncologists. So to be fair, we have also changed our cancer model. We no longer think of cancer as a single mutated clone. We now think of it as a very heterogeneous population of different cancer cell types, and that there is an induction of stroma that's actually very protective of that particular clone that's emerging and involves immune evasion, activating many of the PD pathways, inflammation, and angiogenesis. And you've seen a great example from Gail about how to engage some of these activations. We also have to be aware that many developmental pathway can be reactivated by activation of the stroma. So if one thinks about these heterogeneous population, and someone during the lunch discussion actually said, well, it's a moving target and we keep chasing our tails because each time we apply an agent that's targeted, the new population emerges. And I agree. We can also select very drug-resistant population by using MTD because we kill those cancer cells that are sensitive to that population and emerge new one. This is from a publication in Science Translational Medicine, and it actually argues that if you drop the paradigm of maximum tolerated chemotherapy, you may actually preserve the ecological system within that cancer that has both therapy-resistant and therapy-sensitive cells, and you can engage some of those inflammatory and immune defense cells. When you do maximum tolerated chemotherapy, you definitely get better tumor shrinkage. But that tumor shrinkage may be due to killing everything that can be killed and retaining everything that is going to emerge. So metronomic or adaptive chemotherapy, as Dr. Gaddenby's paper had suggested, it may be a better approach. This is a case in point. I'm a clinician. I always take it back to the clinic. This is a child who presented with blast count. She has ALL, acute lymphoblastic leukemia, with MLL mutation. This is the one time that we do not do well with leukemia. We were able to reduce, and if one can go to 0 and there is no residual disease, our maintenance therapy will prevent recurrence. And that's where our 90% comes from. If we can eradicate the tumor up front, we're good. We've cured, we follow the child, and she does well. We don't do well when there is minimal residual disease that actually recurs. We're able to bring them up with much higher doses of chemotherapy, but then happens that once they relapse, they've relapsed with the most resistant of clones. And you get multiple rounds until the child dies of disease. This is the child that somewhere here may have benefited from genomic analysis, and lestaurinib, which is actually directed towards MLL mutation, was never used in this child. So I hope I suggested that there are some patients that would benefit in this particular case. I am a believer, I do admit, and I believe that it holds a particular promise in oncology because there is way more agent than in other areas of medicine. It is applicable to many other areas of medicine. And in fact, our rheumatologists and our gastroenterologists do a much better job of using these agents. So I also believe that there is an individual specific tumor genomic signature that is actually a conglomerate of those genomic pressures of the host and of the tumor. And that a biomarker-guided treatment is an approach that may not only improve outcomes, but contrary to what's believed, also improve the cost effectiveness of our therapy so that we don't apply futile therapies in these multiple rounds of treatments. I also believe that targeted therapy may be less toxic. So what do we have in 2016? We actually have a lot. So if this is DNA, RNA, protein, and the cell, we actually have a lot of ways of testing our molecular targets. We have genomics, we have traditional in situ hybridization, we have dot plots, we have PCR, we have gene panels, and we have whole genome sequencing. One of the points I was trying to make at the lunch discussion is that all those DNA panels and what I call a candidate-specific approaches will answer questions you already know. So if a basic science supports EGFR in breast cancer, you already know that. What you are looking for are molecular signatures you haven't anticipated. So you can also go for RNA transcriptome. And this is particularly important for those mutations that are developmental pathways that have been activated at RNA level. Most of angiogenesis is not due to DNA mutation. It is due to amplification of pre-existing developmental pathway. Even though angiogenesis is silent postnatally, it actually gets reactivated. Similarly, protein-based molecular targets, we have immunohistochemistry, but our labs in clinics don't have the antibodies. So we don't do them. We also can do Westerns, but there's no clinical lab that will do it in a CLIA-certified lab. And we have CELDI and MALDI, and there's no place to send your patient samples. So proteomics has not entered the clinics. And then there is metabolomics. There are phosphorylation patterns, also possible to do in any research lab. You can do a phosphorylated protein very easy, much easier than a clinician can. You can do methylation patterns. You can do lipid patterns and glycosylation patterns. We cannot, clinicians cannot. So we also have to realize that when we as clinicians send out a genomic analysis, it looks for these, it looks for these syndromes, for Leaf-Romani syndrome, for Lynch syndrome. It doesn't, at best, it uses the candidate gene approach. But very few times you actually get an open-ended approach. And this is what Bill Sellers was talking about when you actually try to give bioinformatics information to a clinician. So the whole genome sequencing is a research tool, and it identified the most frequently mutated genes in a population, not in a patient. And it is fed, and we created all those different protein-protein interaction maps. For most researchers, these protein-protein interaction maps are daily event. For clinicians, we hardly see them. But this is what we actually have. We can use the specific patient-oriented clinical information, proteomic information, cytochrome information, metabolome information, and we can feed it into our tools, overlay it over the exact existing databases. We have GEO, we have TCGA, those are atlases against which we can analyze our specific individual patients. And we can look at protein-protein interaction network. There's many of them. They're all academically curated. And if they're cross-referenced between each other, you can very easily identify the energetic pathway of these different situations and of these different profiles, genomic profiles, and bring it back to the patient and inform the therapy. I'm going to try and make a small attempt as to whether or not we can do this and whether or not we can include it in the patient data. Because the analysis that one does in clinics usually is a handwritten note on my desk or on Andy's desk or on Jack Urban's desk. We don't actually put it into the EMR, into the electronic medical record. So I'm going to try to say that all these different requirements that precision medicine may need exist. So as I said, we need an EMR that streamlines all the different informational streams and that it becomes part of the medical record. We also have to have the interconnectivity of those data sets that Moonshot 2020 is trying to do. We have to cross-reference our data integrity. These are patient lives. And we have to develop a hierarchy of these genomic alteration in individual tumor sample, not for the population, for that particular individual. And we need to, because many of those biologic agents are chemotherapy sensitizer, lower our doses of chemotherapy. So this is an example of integrating a patient's RNA information. So we have published back a year and a half ago with the team of mathematicians a manuscript that indicated that if you use the patient's RNA information, this is done on TCGA patients that actually have data, and look for interactive molecule and calculate the complexity of the network as an energy pathway, then you can correlate the complexity of the network as calculated as Gibbs energy with the progressiveness of the cancer and the 5-year survival. We have done that for different cancers, and this is again published for more details. And then we looked at 4 patients of low-grade glioma and looked whether similar histology, this goes back to I think Clive's point at the lunch session of developing panels and my argument of looking for unanticipated mutations. So these 2 patients within the TCGA both have the histological diagnosis of oligodendroglioma, but calculating the complexity of the network had identified entirely different targets for them, one being an HDAC as the primary target and one having HSP8 as a target. This also activates HSP, but in a different energetic level. So this is an example. This is an example of acute myeloid leukemia, again from the TCGA, from the Cancer Genome Atlas, and it identifies these subnetworks. We call them energetic subnetworks within the large furball of protein-protein interaction networks. And you can see that there are 3 proteins that hold this pathway that are connected with a FLT3, a very known target in AML. And you can see that by inhibiting this, you not only thin this complexity, you separate the activating pathway. Whoever wants to talk to me more about it, just in time frame, I'm happy to talk about how this is done. This has now been automated and computerized using computer tools. So this is another case in point from clinics of how the patient's profiles evolve. This is a child who's 11, diagnosed with glioblastoma in 2011 with a large parietal lesion, and she presented with left-sided weakness. She suffered hemorrhagic stroke, her hemiparesis never resolved, and she went on 2 sets of standard therapy. She went on the ACCN, which is a high-dose chemotherapy trial, in combination with vorinostat, which was the Arm A investigational agent. She was then followed with Avastin temozolomide, and I know there are not friends of temozolomide here, but she did quite well for about 10 months. So she progressed. So we went back this time and looked at her molecular profile. This is a Foundation Medicine panel that has about 600 different genes that have been considered by Todd Golub and Eric Lander, who are the founders of that organization, to be important, clinically important oncogenes. And you can see that this child not only has an F1, which we then found she has systemically, she also has BRCA1 and SOX2. So because SOX2 amplifies Wnt pathway, we had started her on combination of sirolimus and sorafenib with a low dose etoposide. She did quite well for about 4 years. And then we found a newly enhancing lesion on an entirely different place, which didn't look like glioblastoma. It looked like different tumor. And I said, can we get a tissue? So we got tissue and sent it for reanalysis. And this was an important point. She still had the majority of her previous mutations, but she now had an entirely different set of a clonal proliferation. And look at the histology. So her histology changed. This is a classic H&E of glioblastoma with the sort of hemorrhagic regions, the blue round cell tumors with about 30% Ki67 and a very strong GFAP, which is a glioblastoma multiforme marker. Now, note that this is more of a neuroectodermal pattern, more PAT-like pattern of cell differentiation with a lot of more larger cells and a lot less hemorrhagic phenotype. The Ki67 is now in the 67%, and she lost her GFAP. This is no longer a glial tumor, and that is confirmed by the change of her phenotype. So this time around, we know that GLI is activated, so sonic hedgehog is therefore activated. So she was on ceritinib, trametinib, and sirolimus, and she's back to school and dance lessons for a little while. I don't know how she'll do ultimately. We are indeed chasing our tails. So going back to what we will need, and I agree that we will need a different clinical trial design, because if you want precision medicine and individualized therapy, we cannot have populational studies. But this is what the clinicians are faced today. So we get this furball of protein interaction networks, and we will also need what our air pilots need when they consolidate their information, what our nuclear stations need to coordinate their information. We need computers to analyze this for us. The only way we're going to do it is to automatically process the varied resources. We have a lot. Our scientists have been working for the last 60 years on these molecular networks. And these complex socio-technical systems that I'm suggesting will provide a guidance, a technology, individual and teams regulated mathematical or computerized algorithms that will allow you to do it, bring it to the tumor board, discuss it, and provide care. So this is our flow, and again, it's published. If we consolidate all the tests on a particular patient, submit it to computers, recommend pathway therapy, review in tumor board, provide therapy, collect the outcomes, which is something we do not do. And I hope that the infrastructure that Moonshot 2020 will provide will be possible to do, and then bring it back to inform the next decision. So I'm actually quite proud to be part of this initiative. That Floating Hospital for Children is one of the founding members, and I hope we do collect the 20,000 children molecular profiles, and that we will be able to do it. So this is my summary slide. I do believe this is a transformative time in oncology. I do believe that we will need a lot of we will meet a lot of challenges, and both in quality of our tests, in quality of our genomic information, in the quality of interpretation of that genomic information, and that we do need to collect our outcomes, not as a case one study, because unfortunately, all of the case studies out there are the positive case studies. They're not the ones that failed. They're all the good outcomes. And I'll leave it at that, and I'm happy to take questions.
There's something I missed in your talk. Can you explain how you classify cancers in thermodynamic terms?
So,
Because I don't usually hear gives free energy.
right. So our premise is that cancers are not typically following the tissue of origin. They typically follow the biological events that have occurred in that particular population and then ecodynamically suppress or enhance a particular population within the tumor. So if one assumes that a cell is a space in which everything has a certain amount of entropy, certain amount of energy, then if you suppress this, this will come up. So all you need to do within that protein-protein interaction network of that particular patient is to identify the highly energetic pathways. That is a method that the mathematician introduced me to that is actually preordained. You can calculate entropy, and you can calculate Gibbs energy. So that's the term the mathematicians use. We have retained it so that people know which formulation we have used. We have then been able to connect in real biological data out of TCGA that this Gibbs energy correlated with survival. So the higher your Gibbs energy, the poorer survival you had. Yeah. And then we went on and connected, so there is another manuscript where we call it a Gibbs homology, which also, and that's coming out next week in Oncotarget. That incorporates not only the energetic, the physical interaction levels between the different proteins, that also connects the number of connections that the particular protein has. So there are some proteins that are hubs. Some of them were mentioned in the panels. So the RASes, the BRAFs, the ALK1s, they're all hubs. They're busy and they're very interconnected. And that information needs to be brought into the energy information as well, which amplifies your selection of the target. And you can calculate by inhibiting the different proteins, so all thousands of proteins are inhibited one by one, and it tells you inhibition of which protein actually provides you with decrease of that complexity. So that's not done manually.
Good question. In the interest of time, let's conclude this session. Thank you, Dr.